Hello, my name is Brenda Bermudez,
And I'm Ken Wheeler.
and we're part of the Scotch Lab at the Biodesign Center for Environmental Health Engineering. In Scotch Lab, we're studying collected water waste samples that may contain a virus. The genetic information of the virus is examined to determine any changes in its building blocks. Our mentors are Dr. Scotch, an assistant dean of research and professor of biomedical informatics, and Dr. Tope Pillay, a postdoctoral research scholar. They're interested in the evolution of viruses and how they may spread among populations. This research can create new preventative measures and ways to control disease. The virus we're focusing on is canine parvovirus, a strain originating from FPV that affects dogs.
To reduce the possibility of contamination, we clean our workstation thoroughly before and after each use. Typically, our experiment can be split into 2 consecutive days. We do 2 rounds of PCR, a reaction that amplifies the DNA of a sample. Several processes are the same between days and consist of pipetting, centrifuging, and running the samples in the thermal cycler, which controls the temperature needed for DNA amplification.
On day 1, we mix our DNA samples with a master mix that helps conduct the PCR reaction. Items in the mix, such as primers, are used to target a DNA sequence and act as starting points for DNA polymerase to start the replication process while in the thermal cycler.
When we come in the next day, many of the procedures stay similar to the previous ones. We use the resulting samples with a different master mix that targets a smaller, more specific sequence of DNA on the sequence from day 1 during the second round of PCR. Once the thermal cycler step is reached, we run a different program that takes much less time than the one during day 1. Agarose gel is prepared so that we can run gel electrophoresis on the samples. This uses an electric current to pull the negatively charged DNA through pores in the gel, which separates DNA fragments by size, so smaller fragments move through the gel quicker. Using DNA ladders and controls for reference, after dyeing the gel in gel red, we view the gel using a gel dot machine. The resulting image of the gel allows us to determine the presence or lack of targeted DNA. PCR amplified the virus sequence if it was in the sample, which by viewing the gel can reveal if the sample is positive